Hello, everyone. I'm Lori Hutchison, the Executive Director of the Lobular Breast Cancer Alliance and a patient advocate. And I am so happy to welcome you to this advocate chat discussion with the first recipient of the LBCA Deb Mueller Foundation Lobular Breast Cancer Research Grant. In a moment, we're going to hear from Dr. Eugenio Dopeso, who will share information about his study, his award-winning study. Entitled Deciphering Molecular Mechanisms and Therapeutic Vulnerabilities in RHOA Altered Invasive Lobular Breast Cancer. He's also going to tell us about his work with patient advocate Dr. Gitta Jorgensen in developing the application and his plans for working with her going forward. And after he shares information about his study, we'll also hear from Gitta, who assisted him. After their presentations, we will be taking questions off screen. So without further ado, Dr. Dopeso.
Okay. Thank you. So I prepared a few slides to show you. Let me see. Yeah. First, I don't think I have to show you so much about invasive lobular carcinoma, but let me make a small presentation. It's the most frequent special histologic subtype. Probably it's like 15% of the breast cancer that are diagnosed. They have a distinct phenotype, like it's very discoessive with the few cells embedded in the tissue. Of course, they're characterized by CGS1, the allelic activation that cause the loss of expression of a coloring, the cell attachment protein, as you can see here in this image. But however, there is a need for identifying predictors for the progression of the disease and how the tumor is gonna respond to treatment. So of course, it's necessary to invest in the research to try to find these predictors and make all the easier for the oncologists and for the patients to know how the treatment and how the progression of this type of tumors gonna go. So in our in MSK, we have this big data and this big course of breast cancer that are sequencing with our MSK impact panel, and they allow us to investigate a lot of tumors. And we observe here that is in a nice percentage of invasive lobular carcinomas, primary and more frequently in metastasis, we find that RHOA, this gene, RHOA, is important for invasion and migration and the structure of the cells that is already reported, and I'll tell you a little more later, is mutated in a nice bunch of cases. And of course, this happens to be correlated with TDH1 mutations. So making this a clear invasive lobular carcinoma tumor. So that we observe here is these mutations at the molecular perspective. The mutations are very frequently, there's always almost the same mutation located in the same place. And this is important because these mutations in this gene was already in the past described in another type of tumors. Mainly these mutations that were found in lymphomas and it's a very powerful driver of different kinds of lymphomas, and also was described, for example, in diffuse gastric cancer that also have mutations in this gene. And curiously, that we observe in diffuse gastric cancer is that diffuse gastric cancer have a very similar phenotype of lobular carcinoma, being very, as here they say, this cohesive is diffuse and also have loss of expression of a covering. So we think that here be a connection because there is a lot of studies in diffuse gastric cancer where RHOA mutations are also a very important driver for the prognosis and the progression of the disease. So here we check and we compare the ROE mutation lobular carcinomas against a curated group we have in our lab that have not ROE mutations, that also invasive lobular carcinoma, and we observe that significantly that the progression of the disease is faster with lower survival in the case of the patients with ROE mutations. Also, in case of patients only with primary, and also with patients that have metastatic tumors. So also here also I show you that, for example, when we see, especially in the metastasis, in this case, that we observe a curious correlation that the histology of ROE mutation with ROE mutation in the metastatic setting have a high incidence of pleomorphic invasive lobular carcinoma. That is a special subtype of invasive lobular carcinoma that is correlated with a poor prognosis that in invasive lobular carcinoma classic. That is the more common type. That's also, it's already very interesting results, and we're going to try to explore this more in the future. So, because there's very few known why some invasive lobular carcinomas are pleomorphic or classic and drive risk. And also, we observe that the invasive lobular carcinoma with ROE mutations have more cases of triple negative. And of course, that normally the invasive lobular carcinoma are ER positive, HER2 negative, but in the case of ROE mutations, we observe an increase of triple negative breast cancer phenotype. And the triple, as probably you know, triple negative breast cancer is also have poorer prognosis than ER positive or HER2 positive, because there are less type of treatments, for less options for this type of tumor. So that's very important that we try to explore with this. And so, as I tell you now, here is a very summary of the results that I told you, that tumor with ROE mutation show higher incidence of pleomorphic invasive lobular carcinoma, with 65% of them be pleomorphic when compared to the control group. The metastatic tumor with ROE mutation show higher frequency of triple negative breast cancer receptor status when compared to the control group. And also at the level of genes, we observe that ROE alter primary invasive lobular carcinoma metastatic show a higher increase in alterations in chromatin remodeling genes, but still we need to check what is the importance of this. But normally it's also correlated with maybe a poor prognosis or why these tumors are also more aggressive. But we have to keep studying. So basically, the aim of the project we present is, in general, is to investigate the oncogenic role of ROE genetic alterations in invasive lobular carcinoma, considering its established involvement in diffuse gastric cancer, both of which share a discrete phenotype, and explore the potential therapeutic sociabilities associated with ROE alterations in invasive lobular carcinoma, and novel therapeutic approaches for these cases. So that will be, of course, a very interesting thing, as there is not so many specific therapeutic approaches for invasive lobular carcinoma with ROE mutations, or even for invasive lobular carcinoma. So here are the specific aims we write in the grants. Of course, aim 1 is determine the functional impact of ROE inactivation in invasive lobular carcinoma cell lines, cell models. So really validate that these findings I explained you right now are sustained by the research data. And the aim 2 is to elucidate the effect of ROE inactivation on sense signaling in invasive lobular carcinoma, that allows us to explain why, in the cases, why this ROE is important for the lobular carcinoma progression. And finally, determine the therapeutic vulnerabilities of ROE alter invasive lobular carcinoma. So the first is in create these cell models. We work with lobular carcinoma cell lines models. We already previously established and really are very used among all the community of invasive lobular carcinoma researchers. There's these two cell lines, and we try to study the impact when we modulate the levels of ROA inactivation to see what is the changes in growth, migration, invasion, and survival of the cells when we modulate ROA levels to validate that the ROA really is a very important driver in the behavior of these cell lines. In the M2 is elucidate the effects of ROA inactivation of cell signaling. For this, you use a transcriptomic analysis, where we check all the full transcriptomic of the cell line of the cell models we produce. We will produce in M2, and this also this will allow us to identify the pathways that are altered in our models, explain at the level of molecular levels why this the ROA, what is ROA changing in the cell to to promote the more invasive behavior. And finally, using the results from the M1 and 2 to do a drug screening with a library of selected drugs that will be introduced, including compounds targeted in pathways found to be altered upon ROA inactivation in M2. So that will use this with our platform. We use a single drug screening to check what is the drugs they are more effective against the ROA knockdown cells, and through machine learning, we predict we have a machine learning-based prediction analysis, and even study what be the synergies and what is the best use of drug single or in combination, and test these drug combinations and find the more efficient compounds that can target these cells. What is the expected findings? Well, this is a little I think imagining the half of the results, but of course, I expect that we are able to achieve some of these results, and we expect to identify ROA mutated in vesical lobular carcinoma as more aggressive subset of vesical lobular carcinoma. That will be important for the diagnosis of the patients and also maybe for the treatment of the patients. That ROA mutation of loss is anticipated to enhance the invasion and motility of the vesical lobular carcinoma, when to uncover pathways associated with more aggressive tumor behavior. This also is important because maybe we are able to find the pathways and not only be associated with pathways to the ROA mutations, or if not to other changes of mutations or other changes in the cells that are not specific of ROA, and also can explain also why other vesical lobular carcinoma are more aggressive than others. So this is very important for establishing the therapeutic approach. And lastly, we expect to identify therapeutic vulnerabilities linked to the ROA expression or the changes of these molecular pathways. And this is the end. That's why I'm waiting for your questions. Sorry, I won't get this too much specific, but that I can explain you, and happy to receive your comments and questions.
Well, thank you so much. Before we go to questions, though, can you talk a little bit about working with the patient advocate in developing this, and your plans for how you're going to check in and periodically work with Gitta going forward? And then we're going to hear a little bit about from Gitta, and then we'll have questions.
Yeah, sure. So yeah, it was a pleasure to work with Gitta. So she was super helpful to give us some more insights and everything, like we have to, what is have to be our focus, and where we have to focus in one part of the research and the other one, because of course to be more patient focused, and of course to address some of the concerns of the patients in the future. Of course, sometimes research and patients are, sometimes we are disconnected, but happily Gitta was able to help us to make that connection. Of course, we we address that we're gonna make periodically meetings. We check the results. Where of course they're making progress reports and everything to see we are going and we are going in the right direction, and really how we're gonna address. Of course, if there is some troubleshooting, do we have to focus, and we can discuss with her periodically the idea, and of course, when we start to getting some results, of course, we gonna meet and of course writing abstracts for Congress. Of course, we expect to go to have a nice report show in San Antonio next year, and also for the draft of the manuscript and all the steps around. So, but basically, yeah, we start with periodically progress reports, and see what is advanced, and later, yeah, we can move to more show the results in Congress, and of course the publication.
Great, thank you. So Gitta, can you share what your experience was like with?
Yeah. So yeah, sorry. Really interesting. I love, I do a lot of grant reviews as well. I love being involved in the research process. So yeah, what I was kind of focusing on to begin with is things like the lay abstract, making sure everything is very accessible to a lay audience or a patient population. And that's kind of like what I'm hoping to continue. It's a bit like Jose said, you as the patient advocate, you're kind of like the link between the research that's going on. Sometimes, on basic level, translational, it's not always easy to see the link to the patient, but that's kind of why I feel like I'm here as well, to provide that link. And so I think what I would really like to do is also just kind of demystify, that whole process a little bit. So what happens? What do the patient advocate do? And this is not just so that other patient advocates know how to do it. It's actually very much also to encourage researchers to work with patient advocates and sort of demystify it from that perspective as well. So I think it's really exciting, and again, I'm very open to suggestions. I'm big on communication, so I want to be involved as much as possible, and I'd love to get feedback during the whole process as well from any patients who are involved.
Thank you so much, Gitta.